Coming to our next topic, carbohydrate metabolism, glycogen metabolism. So, first going to details of glycogen metabolism, we'll discuss in brief about glycogen. So, what is glycogen? Glycogen is a homopolysaccharide. It is a polymer of glucoses, alpha D glucose units. They polymerize to form glycogen. Glycogen is a animal storage form of glucose. So glucose is stored in our body in the form of glycogen. So the main organs where it is stored is liver and muscle. So these two are the major organs, main organs in which glycogen is stored. So glycogen metabolism involves two processes. One is glycogenesis and the other one is glycogenolysis. First we'll see glycogenesis. What is glycogenesis? Glycogenesis is the formation of glycogen from glucose. So you are synthesizing glycogen from glucose is called as glycogenesis. Right? So glycogenesis, genesis means synthesis. So you are synthesizing glycogenesis. So that's why we call it as glycogenesis. Coming to reactions. So here we'll start with glucose. Now glucose is converted into glycogen, so we'll start with the glucose in glycogenesis. So glucose is converted to glucose 6-phosphate whenever it enters into the cells. So here glucose 6-phosphate, now it is utilized in the synthesis of glycogen. So glucose 6-phosphate in the next reaction is converted to glucose 1-phosphate by the action of enzyme mutase. You are shifting the phosphate from 6th carbon to the 1st carbon, so simply by the action of enzyme called as mutase. And what happens to that glucose 1-phosphate which is formed from the glucose 6-phosphate? So that glucose 1-phosphate is now activated to UDP glucose. So by the action of enzyme UDP glucose pyrophosphorylase. Here UTP and glucose 1-phosphate they react to form UDP glucose, and the enzyme is UDP glucose pyrophosphorylase. So this UDP glucose is the active form of glucose, and this is the form in which glucose units are added to the growing chain. Now this is important compound. Now what happens to UDP glucose? So this UDP glucose is utilized in the synthesis of glycogen, and here we require a pre-existing molecule of glycogen. We call it as glycogen primer. So there is some pre-existing glycogen molecule, we call it as glycogen primer. So whenever glycogen is completely utilized, then a protein called as glycogenin is utilized as the glycogen primer. Right? So there should be some pre-existing glycogen. To that, these glucose units are added by the action of enzyme called as glycogen synthase. So glycogen synthase catalyzes the addition of glucose units from UDP glucose to the glycogen primer. So the key enzyme of glycogenesis is glycogen synthase. Now that glycogen synthase sequentially adds glucose units and it forms alpha 1 to 4 glycosidic linkage. So after a number of glucose residues, that is 8 to 12 glucose residues are added, then after addition of these 8 to 12 glucose residues, then another enzyme comes into action that is called as branching enzyme. The function of branching enzyme is after addition of 8 to 12 residues, some of the glucose residues are transferred to the other chain and it forms the branches. So branches are created by an enzyme called as branching enzyme. So that is the function of branching enzyme, and those branches are created, and the branch point where the branch is formed is alpha 1 to 4, alpha 1 to 6 linkage. So in the linear chain we have 1 to 4 linkages, whereas at branch points, so that is created by the branching enzyme, only at the branch point we have 1 to 6 linkages. Again UDP glucose units they come, and the incoming glucose units are added to the chain by the action of enzyme glycose synthase. Again chain elongation occurs and branching occurs by the action of branching enzyme. So that one more branch is created. Again elongation of that branch occurs, and again by the action of enzyme glycogen synthase, and by the action of enzyme branching enzyme branches are formed. So elongation is brought by glycogen synthase, branching is done by branching enzyme. So these two processes will continue to form a very compact molecule is called as glycogen. So this is how glycogen is synthesized from the glucose by the action of a key enzyme called as glycogen synthase. So the key enzyme of glycogenesis is glycogen synthase. So the rate of glycogenesis depends upon this particular enzyme. Coming to regulation of glycogenesis, how this glycogenesis is regulated. So this is brought by two mechanisms, that is glycogen synthase and the other one is hormonal regulation. So as I told you, the key enzyme of glycogenesis is glycogen synthase. Whenever you add a phosphate group, the activity of glycogen synthase decreases. So covalent modification, that is phosphorylation, decreases the activity of glycogen synthase. So this is how the glycogenesis is regulated. So whenever you remove the phosphate group, dephosphorylation, the activity of glycogen synthase increases. And coming to another way in which it is regulated is hormonal regulation. In case of hormonal regulation, we'll see there are two sets of hormones so that which affect the blood glucose level. So one set of hormone, they increase the blood glucose level, and the other types of hormones, they decrease the blood glucose level. The only hypoglycemic hormone is insulin. So it always tries to decrease the blood glucose level, whereas glucagon, it tries to increase the blood glucose level. So here glucagon increases the rate of expression of all the key enzymes of expression of the key enzyme of glycogenesis, that is glycogen synthase. So it increases the activity of glycogen synthase so that overall glycogenesis can be increased. So this is the effect of hormones and their effect on the regulation of glycogenesis. So covalent modification increases the activity of glycogen synthase by dephosphorylation and decreases the activity by phosphorylation. So hormonal regulation, it is brought by two sets of hormones, glucagon and insulin. Insulin always tries to decrease the blood glucose level. To decrease the blood glucose level, it has to increase the utilization of glucose. So insulin converts glucose into glycogen, so it increases the glycogenesis. Whereas glucagon, which always tries to increase the blood glucose level, it decreases the rate of glycogenesis. So these two hormones, they have opposite effect. One increases the glycogenesis, which tries to decrease the blood glucose level, and the other one decreases the rate of glycogenesis, which tries to increase the blood glucose level. So this is the regulation of glycogenesis. Now coming to the another part of glycogen metabolism, glycogenolysis. So what happens in glycogenolysis? So we have said in case of glycogenesis, that is formation of glycogen from glucose, here reverse occurs. So glycogen is converted into glucose. So the breakdown of glycogen to glucose is called as glycogenolysis. So you are lysing this glycogen into glucose. You are breaking this glycogen into glucose. So this process we call it as glycogenolysis. So in case of glycogenolysis, we'll start with a glycogen molecule. So that glycogen molecule having all the glucose units linked by alpha 1 to 4 glycosyl linkage in the linear chain, and at branch point 1 to 6 linkage they have. So all those glucose units now they are released from the glycogen molecule in glycogenolysis. So the first reaction of glycogenolysis is catalyzed by the enzyme called as glycogen phosphorylase. So this is the key enzyme of glycogenolysis, glycogen phosphorylase. The action of glycogen phosphorylase is that, so whenever it acts on glycogen, it releases one glucose unit in the form of glucose 1-phosphate. So that is the effect of glycogen phosphorylase on glycogen. So it releases the glucose units as glucose 1-phosphate. Now, but it has some restrictions. So glycogen phosphorylase can act on the glycogen molecule, it can act on a branch, but it can remove glucose units from the branch point, leaving the 4 residues. So it cannot act on the glucose residues from the branch point. So leaving 4 residues from the branch point, it can remove all the glucose residues as glucose 1-phosphate. So that is a restriction of glycogen phosphorylase. Now, once glycogen phosphorylase removed all the glucose units, leaving 4 glucose residues from the branch point, then another enzyme comes into action that is called as debranching enzyme. So this debranching enzyme has two functions. One is it has transferase function, and the other function is it has glucosidase function. Now, the transferase function of debranching enzyme is, so out of 4 residues which are left from the branch point, it transfers the 3 residues to the another chain, right? So that is the transferase function of debranching enzyme. So once it transfers the 3 residues out of 4 to the linear chain, so there is only one residue at the branch point linked by linked by alpha 1 to 6 glycosidic linkage. Now what happens to that glucose residue, single glucose residue at the branch point? And that is removed by the action of enzyme called as glucosidase. So debranching enzyme has two activities. One is transferase action and the other one is glucosidase action. So glucosidase action of debranching enzyme, it removes the glucose residue at the branch point as free glucose. It is not released as glucose 1-phosphate, but it is released as free glucose. So now we have only a linear chain. Again, glycogen phosphorylase comes into action and it releases all the glucose units as glucose 1-phosphate. So now we have glucose 1-phosphate, and those glucose 1-phosphates, they are converted to glucose 6-phosphate by the action of enzyme called as mutase. So that mutase converts glucose 1-phosphate to glucose 6-phosphate. Glucose 6-phosphate now is further converted into glucose by the action of enzyme called as glucose 6-phosphatase in liver. Whereas in muscle, there is no such enzyme which can convert glucose 6-phosphate into glucose. So the fate of glycogenolysis, the end product of glycogenolysis in muscle is glucose 6-phosphate that enters into glycolysis to supply energy. And in case of liver, that glucose 6-phosphate can be converted into glucose by the action of enzyme glucose 6-phosphatase. So the purpose of glycogenolysis in liver is to maintain the blood glucose level, and the purpose of glycogenolysis in case of muscle is to supply glucose for energy, right, for glycolysis. So this is the reactions of glycogenolysis. Here the key enzyme of glycogenolysis is glycogen phosphorylase. And coming to its regulation, so generally whenever glycogenolysis occurs, the blood glucose level increases. So it supplies the blood glucose level, it maintains the blood glucose levels by converting that glycogen molecule into glucose. Right. So the key enzyme of glycogen phosphorylase is regulated by covalent modification. So whenever you add a phosphate group to glycogen phosphorylase, the activity of glycogen phosphorylase is increased. So this is opposite to that of glycogen synthase. Whenever you add a phosphate group to glycogen synthase, the activity is decreased there. But here, whenever you add a phosphate group to glycogen phosphorylase, the activity of glycogen phosphorylase is increased. So this is how glycogenolysis is regulated. Coming to another way, hormones. So as we discussed, there are two sets of hormones. One increases the blood glucose level and the other one decreases the blood glucose levels. So insulin is a hypoglycemic hormone. It always tries to decrease the blood glucose level, whereas glucagon, it increases the blood glucose level. So here the effect of glucagon on glycogenolysis is it increases the rate of glycogenolysis, whereas the effect of insulin on glycogenolysis is it decreases the rate of glycogenolysis. So these two are the hormones which regulate the glycogenolysis. Right? So this is how glycogenolysis is regulated. So there are some important diseases related to the glycogen metabolism. We call them as glycogen storage diseases. So glycogen storage diseases are because of accumulation of abnormal type of glycogen or abnormal amount of glycogen leads to a condition called as glycogen storage disease. Right? Two things will happen. It may be accumulation of abnormal amount of glycogen or abnormal type of glycogen. And we have some important diseases like type 1 Wong-X disease, type 2 Pompe's disease, Cory's disease, Anderson's disease, McArdle's disease, and Hurst disease, Torres disease. So these are the important glycogen storage diseases. The most important and disease is type 1 disease, type 1 glycogen storage disease, that is Wong-X disease. Here the defective enzyme is glucose 6 phosphatase. So because of deficiency of this particular enzyme, glucose 6 phosphate cannot be converted into glucose. As whenever there is deficiency of glucose, it leads to a condition called as hypoglycemia. So in case of type 1 glycogen storage disease, the type of hypoglycemia is fasting hypoglycemia. In case of fasting conditions, actually glycogenolysis has to occur and it maintains the blood glucose levels. But here, due to defect in this particular enzyme, glucose cannot be released from glycogen, so that it leads to a type of hypoglycemia. We call it as fasting hypoglycemia. That is the important feature of type 1 glycogen storage disease. Apart from that, there is increased concentration of lipids in the blood, and that is in case of type 1 disease. And apart from that, there is lactic acidosis, there is hyperuricemia, and that results in gout. Right? So these are the important features of type 1 glycogen storage disease. Coming to Pompe's disease, that is because of deficiency of acid maltase. And coming to Cory's disease is because of deficiency of enzyme, debranching enzyme. Anderson disease because of defect in the branching enzyme. And McArdle's disease because of deficiency of muscle phosphorylase. And Hurst disease because of deficiency of liver phosphorylase. So these are the glycogen storage diseases and the defective enzymes. Right? And that's about the glycogen metabolism. You have seen what is glycogenesis, glycogenolysis, and what are glycogen storage diseases. Now coming to the other way in which glucose is subjected to oxidation. So we have seen the major way in which glucose is oxidized is pentose phosphate pathway. The major way is glycolysis, and other way in which glucose is oxidized is pentose phosphate pathway. So here the purpose of oxidation of glucose by HMP pathway is it has two roles, that is it supplies the NADPH and it supplies the ribose 5 phosphate. It supplies the ribose which is required for the synthesis of DNA and RNA. So these two are the important functions of HMP pathway, supplying NADPH and different sugars. Like the most important one is ribose, which is required for the synthesis of DNA and RNA. So when you see site, so this pentose phosphate pathway occurs in the cytosol. All the enzymes concerned with this pathway are located in the cytosol, and the most important tissues which are very active are mammary glands, adipose tissue. These are the important tissues which are very active in this pentose phosphate pathway. Coming to phases, there are two phases in HMP shunt pathway. One is oxidative phase and the other one is non-oxidative phase. Yes. Coming to reactions of HMP pathway, so we will start with the glucose 6-phosphate. The glucose enters into cell, it is trapped at glucose 6-phosphate. The same concept here also. So glucose 6-phosphate may enter into glycolysis, may enter into HMP pathway, may be utilized in synthesis of glycogen, maybe it may enter into uronicase pathway. So depending on the cellular requirements, glucose is diverted to that particular pathway. So here, in case of HMP pathway, glucose 6-phosphate is converted to 6-phosphogluconolactone by the action of enzyme called as glucose 6-phosphate dehydrogenase. So this is the first step in which the NADPH is produced in the HMP pathway. So NADPH is a type of reducing equivalent produced by HMP pathway. The important source for the NADPH is HMP pathway. So what happens to the 6-phosphogluconolactone which is found in the first reaction? So that 6-phosphogluconolactone is converted to 6-phosphogluconate by the action of enzyme lactonase. Right? So 6-phosphogluconate in the next reaction by the action of enzyme dehydrogenase, 6-phosphogluconate dehydrogenase So this is the second oxidative step in the HMP pathway, where one more molecule of NADPH is produced, second molecule of NADPH is produced. So here there is decarboxylation also occurs, so this is oxidative decarboxylation. So there is conversion of reduction of NADP plus to NADPH, and also there is removal of one carbon as carbon dioxide. So the 6 carbon 6 phosphogluconate is converted to a 5 carbon sugar called as ribulose 5 phosphate by the action of enzyme 6 phosphogluconate dehydrogenase. So up to these reactions, conversion of glucose 6 phosphate into ribulose 5 phosphate, this phase is called as the phase 1 or oxidative phase of HMP pathway. So in this phase, the important compound which is generated is NADPH. Now coming to non-oxidative phase, what happens in the non-oxidative phase? So there is interconversion of different sugars. For example, so that ribulose 5-phosphate which is formed in the phase 1, now it is isomerized to ribose 5-phosphate and it is converted into xylulose 5-phosphate by the action of epimerase. So isomerase converts ribulose 5-phosphate into ribose 5-phosphate, ribulose 5-phosphate into ribose 5-phosphate, and epimerase converts ribulose 5-phosphate into xylulose 5-phosphate. So this ribose 5-phosphate and xylulose 5-phosphate, they react together by the action of enzyme called as transketolase, and this transketolase requires the coenzyme thymine pyrophosphate, so xylulose 5-phosphate and ribose 5-phosphate, they are converted into C-dihaptylose 7-phosphate and glyceraldehyde 3-phosphate. And these two react again by the action of enzyme called as transaldolase, and that converts glyceraldehyde 3-phosphate and C-dihaptylose 7-phosphate into fructose 6-phosphate and erythrose 4-phosphate. Again, that erythrose 4-phosphate and xylulose 5-phosphate, they react by the action of enzyme called as transketolase, and that is converted into again glyceraldehyde 3-phosphate and fructose 6-phosphate. So glyceraldehyde 3-phosphate may enter into glycolysis because it is one of the intermediates of glycolysis, and the same way fructose 6-phosphate is also intermediate of glycolysis. So basically, in case of non-oxidative phase of HMP pathway, there is interconversion of different monosaccharide phosphates. So that is glyceraldehyde 3-phosphate can be converted into erythro 4-phosphate when it reacts with C-dihaptylose 7-phosphate by the action of enzyme called as transaldelase. So different monosaccharide phosphates are formed in case of non-oxidative phase of HMP pathway. So one of the most important sugar which is formed in this phase, non-oxidative phase, is ribose 5-phosphate. So that ribose 5-phosphate is utilized in the synthesis of DNA and RNA. So that is the significance of non-oxidative phase of HMP pathway. So here you can see significance. So we'll discuss under two headings, that is, what is the significance of oxidative phase and what is the significance of non-oxidative phase. The significance of oxidative phase is it generates the NADPH. So that NADPH is utilized in some important reductive biosynthetic reactions. For example, for the synthesis of fatty acids, for synthesis of cholesterol, we require NADPH. So such type of synthetic reactions, they require NADPH, and it is supplied by the oxidative phase of HMP pathway. Right? Coming to other uses of NADPH, so this NADPH is utilized in the phagocytosis. Right? And the other use of NADPH is it maintains the transparency of the eye. So transparency of the lens is maintained by the NADPH. And apart from that, this NADPH is utilized in the reduction of oxidized form of glutathione into reduced form of glutathione, for the conversion of oxidized form of glutathione, that is GSSG, into reduced form of glutathione, who call it as GSH. So we require NADPH. So that is the most important function of NADPH, which is produced in the oxidative phase of HMP pathway. And coming to non-oxidative phase, we discussed here ribose 5-phosphate, the most important sugar required in the synthesis of DNA RNA, is from the non-oxidative phase of HMP pathway. And coming to G6PD deficiency, what happens in case of glucose 6-phosphate dehydrogenase deficiency, the first enzyme of HMP pathway, which converts glucose 6-phosphate into 6-phosphogluconolactone. So whenever there is deficiency of this particular enzyme, it leads to decreased availability of NADPH. So whenever there is decreased NADPH concentration in the cell, for example, the most affected cells are erythrocytes. So whenever NADPH is low concentration, whenever it is not available, the concentration of hydrogen peroxide increases in the erythrocytes, and that hydrogen peroxide destroys the RBC membranes, and it leads to excess lysis of RBCs, and that is called as hemolysis, and finally it leads to anemia. We call it as hemolytic anemia. So here the lysis of RBCs because of excess hydrogen peroxide, and that hydrogen peroxide has to be detoxified actually by glutathione, reduced form of glutathione, that is GSH. But for the formation of reduced form of glutathione, we require a particular compound called as NADPH. So without NADPH, reduced form of glutathione cannot be formed, and without reduced form of glutathione, hydrogen peroxide cannot be detoxified. So whenever the concentration increases, it leads to hemolysis, and finally it results in a condition called as hemolytic anemia. So that is the significance of G6PD deficiency, glucose 6 phosphate dehydrogenase deficiency. Thank you.